Welcome, everyone, to Stability Hub's webinar, Handling Accelerated Stability Failures. We would encourage you to turn your camera on if possible, but stay muted unless you are asked to comment further on a question. This session is being recorded and will be available in the articles and video section at stabilityhub.com. Today's webinar begins with a presentation and will be followed by answering your questions and more broadly discussing the process of dealing with failures at accelerated conditions. Please use the chat feature to submit your questions as they occur to you, and they will be answered in order after the initial presentation concludes. If desired, you can address your question to a specific panel member. In the absence of your questions, we've assembled an interesting array of frequently asked questions. Our presenter today is Pearl Pereira-Nambiar, founder of RegXpertPRO, where she helps pharmaceutical CMC teams navigate complex analytical and regulatory challenges with greater clarity and consistency using science-backed tools, structured workflows, and expert solutions. Her work focuses on dissolution, specifications, impurity control, stability interpretation, analytical documentation, and deficiency responses. Visit her site at www.regexpertpro.com and follow her on LinkedIn. Our panel members today are Chris Latos, Manager of Stability Services, Hollister Incorporated, a manufacturer of quality medical products for critical care markets. Ken Waterman, Founder and President of Freethink Technologies, Incorporated, a company developing new products and advancing new ideas. Ken is a pioneer in accelerated predictive stability, and you might recognize his product, ASAP Prime. Walter Routh, founder and CEO of Sagana Quality Consulting, is an ex-AbbVie Stability Director and is currently Associate Editor at Stability Hub. And yours truly, John O'Neill, a lifelong stabilitarian, Pharmaceutical Stability Discussion Group facilitator, and founder of Stability Hub. We'll get started with today's program. Questions are welcome via the chat feature throughout the session and will be answered following the slide portion. Pearl, the presentation is now yours.
Thank you so much, Walter, and a very good morning, good afternoon, and good evening to everyone. Before we start the presentation, welcome, and I'll just share my slides.
Perfect.
Yes. So once again, welcome everyone to this presentation on managing accelerated stability failures. Just to set the expectations right at the onset, we will not be reading the guidelines Q1A and Q1E. What we'll be focusing today is what happens after an accelerated result appears. So how do we decide whether it's actually a significant change or what pathway it triggers? And more importantly, how do we interpret that result, what it means for the product and what it means for its proposed shelf life? Because this is where we often get stuck. We kind of overreact when we see such kind of results during the accelerated stability and think that probably the shelf life of the accelerated result has failed. Or on the flip side, it could also be construed as the lifetime results, the lifetime long-term stability results anyways, they comply. So it's just a failure in the accelerated stability. So it's not a big issue altogether. The point is, neither conclusion is necessarily correct here. So today what we will be doing is we'll be walking through the regulatory pathway where we'll also spend time on the scientific questions that sit behind this thinking. We'll start with a simple example here. This is a test for assay where you have a shelf life specification of 90 to 110% of the label claim. The initial result, as we see here, is 100.0%, and we also have the 6-month accelerated result of 94.8%. So the question here is, would you report 94.8% as an acceptable assay result? So most of us would say yes, of course, because it does comply with the predetermined or the approved specification of 90 to 110% of the label claim that had been set. However, this is only one part of the assessment because as we see here, the result has changed by 5.2% with respect to the initial value. So even though it remains within the specification, it is still considered as a significant change under accelerated stability condition. It meets the ICH Q1A definition of a significant change. So this distinction between an out of specification, a result that meets specification, however, is still a significant change under accelerated condition. This is the starting point of the discussion we'll be having today here. So the key point here is a result can be acceptable. It can be within the specification. However, that can still trigger an investigation because that is that is construed as a significant change under accelerated stability conditions. And what happens here, it will change the regulatory pathway for shelf life evaluation. We have two questions here. The first one is, does the result meet its acceptance criterion? And when you look at the second question, it asks, does it meet the ICH definition of a significant change? Now, both these questions have different purposes. Compliance to a specification tells us whether an individual result is acceptable against the predetermined acceptance criteria. On the other hand, the significant change tells us whether the stability result, how a stability result should be evaluated. But that doesn't mean that one cancels the other. They are both complementary. And when you look at it on a slide in terms, when you segregate it as two different questions, they appear obvious. But in real time, when we do work in the laboratory, it's often very common to confuse the two. And usually what happens is the review stops when you have an out of specification result, and it's very easy to conflate both the concepts there. What we need to do is we need to keep the two assessment separate, and out of specification a result has different consequences versus a result that is deemed as a significant change under accelerated stability condition. That is a different outcome, and it needs a different regulatory treatment with respect to the stability shelf life evaluation. Before we move on, let's look at what constitutes a significant change. For a drug substance, that's pretty straightforward. It's just that failure to meet its specification at any interval is considered as a significant change at any interval during the accelerated stability conditions. That's a significant change. For the drug product, it's kind of different. Here, the definition here is specific to the attribute of the drug product. You have a one for assay, as we saw in the example earlier, a 5% change in assay from its initial value or failure to meet the acceptance criteria if we are working with biological or immunological products. That is considered as a significant change. Now, the operative here, a word here is initial value. It's not that you have any higher value, say, for example, taking the same example as we had seen earlier, where we had initial value of 100.0%. And maybe at the next interval, say at 3 months, you would see 102%. And at that time, if you have another result of, say, 95%, that would not be a significant change because you cannot compare it against the value of 102 because that's not an initial value. So that is what we need to keep in mind when we define what exactly is a significant change. It is always compared against the initial value that is reported. Likewise, even for a test like a dissolution here as well, failure to meet the acceptance criteria for dissolution for 12 units, that is at the S2 stage, that is also considered as a significant change. So it can also happen that the drug product might not comply at the S2 stage and then will proceed to the next stage, that is the S3 stage, and would comply there. So technically it complies to the specification. However, that because it does not comply at the S2 stage, that is at 12 units, this is still considered as a significant change, and accordingly, the shelf life evaluation would therefore change. Then we have other tests like degradation products. You also have the test for here, even in degradation products, any non-compliance to the specification limits that have been set, that is considered as a significant change. Now, here compliance is not only with respect to the total degradation product or impurities, even if a single impurity, be it specified or unspecified in the impurity profile, if it does not comply to the predetermined specification, it will be considered as a significant change. For a test like appearance, physical attributes, and functionality, again, your failure to meet acceptance criterion also is considered as a significant change. However, you need to use a scientific judgment here, because certain physical changes might be expected. For example, a cream may soften, or depending on the exposure or temperature, if it is well above the normal storage condition, a capsule might show certain changes. So, but this doesn't mean that we ignore these changes as expected under the accelerated stability conditions. What this means is that we need to ask a more useful specification. Not that does it comply. Instead, we should be asking is this an expected and a reversible response to the accelerated condition? Or actually, does it reveal the product or the packaging vulnerabilities that we see, that are expected to be seen during the normal storage conditions here? Likewise, pH is another test here. If it fails the acceptance criterion, that is also considered as a significant change. But we do not stop just at the numerical value. What we need to do is still understand what exactly is driving the shift, and whether there are other attributes as well that are changing with this attribute of pH of the drug product. Before we designate any result as, or before applying any regulatory interpretation here as for the significant change that we see under accelerated conditions, we need to confirm whether the observed change, is it analytically valid? Was the calculation correct? Was the method performing as expected? Was the system suitability or sample preparation, was standard preparation, were there as per the acceptable method details? And is there any assignable laboratory cause that could be identified? That could be, it could be dilution error, or it could be due to the handling of the sample, or contamination, any event that would invalidate that result. But again, this confirmation should not come into the way of, explaining away any kind of inconvenient result here. Once you determine that, and there is no assignable regulatory cause here, or regulatory error here, laboratory error here, what this means that this result is now valid. So now at this point, the investigation needs to move from what went wrong in the lab to now what is the product trying to tell us. So we also look at the chamber history, where the sample has been, stability samples have been stored, sample and container integrity, and are there any specific packaging related observations? For example, if there is only a single blister cavity that is damaged, that may be an isolated defect. However, if the same moisture related change appears across several units, or patches, or at different time intervals, now those may indicate a broader product or package sensitivity here. And finally, the result should be viewed the results should be viewed in the same direction of change. So are there other related attributes that are also changing along with this attribute? So this is also what we need to look at. So confirmation is not just here about repeating the test. It is also deciding whether the results are credible or whether we actually need to take that step forward and now look at what is the applicable stability pathway here. Now that we have assigned the right significant change under accelerated stability conditions, let's have a look at the general pathways that are applicable for two types of products. One is the refrigerated, and we will also look at the temperatures that samples or batches that are stored at room temperature. As per the decision tree here, you need to understand whether once you've confirmed that the significant change has occurred within 6 months, in case if no change has occurred, obviously you do not have to take any action. If yes, it has, then we move to step 2. And this is where we diverge between the storage conditions or the stability pathway for a refrigerated product versus the product that has been stored under room temperature. Now, for a refrigerated product, if the timing has changed within 3 months, so here in this case, it does not support any extrapolation here. So the proposed shelf life has to be then based on the available long-term data, and any kind of early accelerated data change also, it raises another alarm because that's a practical question about short-term excursions here. For example, what could happen possibly during a routine distribution or dispensing, transportation, or even temporary storage outside the refrigerator? Those questions need to be answered too. Sorry. And again, in case if there is any change within between 6 to 3 months, here the interpretation could be different from the change occurring between 3 months. But the key point here is that the accelerated data for refrigerated products is not simply a quicker version or a shortcut of the long-term data. Here, the stress can be substantial, of course, related to the labeled condition, but it also gives us the scientific relevance for that change that is observed, and it therefore needs to be considered carefully. Coming to the samples that are stored at room temperature, here again, if a sample is stored at room temperature, you need to keep in mind where intermediate testing is applicable, because there are certain storage conditions where long-term is considered as 30 degrees, 65% relative humidity. In this case, this pathway would not be applicable. However, if you have room temperature at 25 degrees Celsius and 60% RH, in that case, intermediate testing would be applicable. You need to then confirm whether significant change, whether that does occur at the intermediate condition. If it is a yes, then no extrapolation is allowed here. And in this case, the proposed shelf life would be based on the long-term data that is available, and even a shorter shelf life would be required here, depending on the trend that is seen for the data. That is the reason why you need to have a statistical analysis and also to check whether there is any variability in the long-term data that has been reported. In case if we do not, if there is no significant change at the intermediate conditions, you need to look at whether you need to then ask two questions. One is the long-term data amenable to statistical analysis? And was the analysis actually performed? So there are some quantitative attributes, such as it would be easier to do it for an assay where you have numerical values here, statistical analysis. However, if the significant change is for a test like, say, appearance, then in that case, it would be very difficult to have a regression analysis done here. So this is also what you need to consider when you are evaluating it for a significant change, and there is no change under intermediate conditions. In the case if it is backed by supporting data, in that case you can extrapolate it. If it's for a test like appearance, then you add 3 months to the available long-term stability data that is available. And in case if both 1 and 2 questions you have answered a yes there, it's backed by statistical analysis and it does support the relevant stability data. We have a mathematical equation here where you multiply the interval by 1.5, and that number shouldn't exceed your long term with the addition of 6 months. But an important point that we need to keep in mind there, that these are upper boundaries. It is not a free pass to that extrapolating and getting an extended shelf life. What at the end you need to do is you need to support it with the relevant data, and not just for the test you're evaluating, but it will be for all the applicable tests for that specific drug product. Let's look at an example here. We have an immediate release tablet. We have long-term data of 18 months. Accelerated condition shows significant change at 6 months. And intermediate stability does not show any significant change. We also know that the data, when evaluated, it is suited to statistics, and this is not a test like say appearance, so it's amenable to statistics here. Statistical analysis also supports compliance to at least 24 months. So this is what we have seen when we ran stats. So the question here is that, can a sponsor propose 24 months? Now, if we use the same equation as we saw in the earlier slide, we have long-term data of 18 months. We multiply it by 1.5. That gives us 24 months. And we also take the other equation where we have 18 plus 6. That gives us 24 months. So taking the more restrictive criteria, we have a shelf life of 24 months. But does this mean that the sponsor can actually go ahead and propose 24 months? The answer is could be, potentially. Why potentially is that? Because this calculation alone does not prove that 24 months is the actual stability period here. You need to provide that all the stability attributes, including the complete data package, should support this 24 months of shelf life that is being proposed by this equation. For example, if a dissolution test supports only 24 months, then the overall proposed shelf life cannot exceed 24 months, even though we actually see a number of 24 months here. And again, verification. So even after you propose a shelf life of 21 or 24 months, the long-term testing has to continue till the proposed shelf life to prove that actually the experimental results are in line with the theoretical proposed extrapolated shelf life. Now that we have seen the regulatory pathway that is applicable here, that only tells us what route is applicable. The decision tree doesn't tell us the next step. That is why this change has occurred. So an accelerated change could reflect a degradation pathway that is also operating under long-term stability conditions, though it could be at a slower rate. It can also refer to a physical transition that occurs only after crossing certain critical temperatures or humidity barriers. It could possibly indicate moisture ingress or oxygen sensitivity, loss of solvent or water. It may also imply sensitivity to short-term excursions. At times, we also see that accelerated conditions can also activate a degradation mechanism that would not be likely under the expected or relevant labeled storage conditions. But this last explanation, that this observation that we see under accelerated condition that is not relevant to the actual label storage condition. Now, this requires evidence. So we should not label the result as only accelerated condition specific simply because this has complied under the real or the long-term stability conditions. What we need to show is what kind of mechanism is operating here, why this accelerated stress condition has specifically activated it, and why this mechanism precisely is unlikely or whether it is likely to matter under normal stability storage or excursion conditions here. Now, this is where everything comes together. So we need to reconcile the accelerated result with long-term and intermediate trends. We need to understand that whether the degradation mechanism or any kind of physical change mechanism, the mass balance, but here again, we do not simply treat it as a mathematical or an arithmetic exercise. We need to look at whether the drop in assay is compensated by corresponding increase in degradation products. Mind you, there could also be volatile impurities. So all these factors also need to be kept in mind. So the real condition here is whether the analytical method is capable of detecting and explaining the observed change. And along with this, the container closure system should also be a part of the assessment right from the onset and not as an afterthought here. Why? Because was oxidation, if that was the actual mechanism of the degradation, was it caused by the container closure system? Was it because of the headspace that was seen? So this is the kind of discussion that we need to actually put into our dossier. So what Q1E tells us is of the consequence, but the scientific assessment is what will tell us whether the proposed shelf life actually makes sense. So now this is the conclusion that we often see is that the failure has occurred only under accelerated conditions while long-term results comply. So a stronger assessment here is that under accelerated conditions, it's the significant change, it does not actually establish instability under the proposed storage conditions, but you need to still reconcile the results with a likely mechanism. Is it the container closure system, the intermediate and the long-term stability trend, and also the shelf life that is being proposed? So the strength of that conclusion depends on the evidence that is used to support that conclusion. So even if the paragraph is well written, it does not compensate, however beautifully this has been put, it will not compensate for any missing mechanistic data or a stability data that would actually be useful to support the argument that we put forth. So we need to ask 5 questions before we justify any shelf life, propose any shelf life. First is the accelerated result actually confirmed as a valid significant change? What Q1E pathway does is actually applicable depending on whether it's a freeze product, a refrigerated, or a room temperature storage product. And is extrapolation available here? What is the mechanism that this degradation is, or this significant change operates under? And is it expected during the routine labeled storage conditions or during the expected excursions? And is it relevant to this container closure system? How does it reduce or it increase that kind of risk? And does the complete data package, all the data that is there, it supports the proposed shelf life across every relevant attribute, batch and pack? And where extrapolation, if it has been used, how will that be verified? So the most important point here is that long-term stability results comply. Yes, that is an observation. However, on its own, it is not an explanation for an accelerated significant change here. So to summarize, we have 3 key points here. That is how I look at accelerated significant change in 3 levels. First is we have ICH-Q1A, which asks what constitutes a significant change and what additional stability evaluation is triggered. Then the baton is taken over by Q1E, which asks what shelf life and what degree of extrapolation can the available data actually support. And third, to tie it all together, you have the scientific assessment, which asks the most important question: why did this change occur? Is it relevant to the label storage condition that is expected, the container closure system, and the proposed shelf life? So, an accelerated stability, significant change in itself is not automatically any shelf life failure. But again, it is also not a result that can be dismissed simply because the long term stability data passes. It's an evidence that the product or the packaging it is sensitive under accelerated stress conditions. So the job of a stability assessment here is to determine what that sensitivity means for the actual claim for that drug product. And here, the framework that we discussed today has been based on the currently final ISIG, the effect, the one, the effective Q1A R2 stability guidelines. I have not considered the consolidated Q1 revision because that is still in the draft. So I have not treated the draft concepts here. I have only considered the one that has been effective. So thank you. With this, we conclude, and thank you so much for your time and attention. I'll be very interested to hear your questions, comments, and experiences, particularly situations which you would have faced in the lab regarding to the topic that we have just discussed. Thank you.
Thank you, Pearl, for undertaking this topic. It's been long underdeveloped in our experience, and we're glad that you tackled it for us. As we wait questions for come in from the audience, I can ask you if you just mentioned about time points are valid in many cases, even though there might have been a significant change. Is there a compliant basis for canceling a time point or an accelerated station in anticipation of a failure? In other words, if you had a couple of batches that legitimately did fail, are you familiar with any reason not to cancel the last of three batches that you anticipate the failure to occur, or does that bring on regulatory problems?
Again, that depends because if you have already you need to have that data to you need to have scientific assessment here first to justify. So just you cannot do away with you can't do away with any kind of stability interval just like that because you anticipate a failure here. You need to support it by the actual data and why you expect that failure. So to that kind of explanation and rationale has to be a part of the explanation that we provide. So you just can't do away with an interval because you expect to see a failure there.
Okay. And we have a question from the audience. If it's within the bounds of the scope of our topic, she'd like to know how historically has the definition of significant change being only 5% come about when some products have a potency range at release of 95 to 105 or broader. So is that the nature of the product that we're speaking of here, or do you have a experience with knowing where that definition came from for those kind of products with a wide potency range?
But keep in mind, this is potency change.
Mm-hmm.
So as long as your content uniformity and your sample size is enough, it's easy to detect a 5% change.
Okay, thank you.
For most products.
So I have a question.
Statistically, that makes sense.
Thank you, Ken. I have a question for Chris Latos, as one of our practitioner stabilitarians here today, followed up by Ken, who would be able to comment on that as well. So, Chris, what, when, and how do you take into account a stability chamber excursion? How significant does that have to be in your world before you start looking at the impact on the product?
Yeah, so especially in the case of an accelerated chamber, we use continuous monitoring. So when we get an alert if the chamber does have an excursion, and then per our SOPs, if it's out of range for longer than 24 hours, we can do an assessment. Typically with accelerated, the common practice is, see what temperature see how what the range of the deviation was and the duration of that deviation. Again, if it's over 24 hours, we'll act on it. In some cases, samples could be transferred to another stability chamber. In other cases, we may just simply add additional time to every study that's impacted. So all the studies that are in that particular chamber, we may add, if the chamber went down for 2 or 3 days, we may add just an additional 2 or 3 days to the poll intervals. So that's typically how we handle those.
Okay, and then Ken's part. Thank you, Chris. Ken's part, once characterized for your product, is it easier to determine the impact of these stability excursions on the stability budget? In other words, you extensively study your product ahead of time. So I think it may be easier to predict what the impact is statistically. Going back to our discussion once where we had a regulatory question, the inspector asked, what's the cumulative impact of several excursions, none of which actually were out of range? And
Well,
I think
why
the
don't we
response was that you could determine
let's take a step back. First, you're dealing with out-of-spec chambers, and even in-spec chambers, one of the biggest problems we have is the allowed range is quite large. So if you, for example, are supposed to be refrigerated, your temperature could range anywhere from 3 to 8 degrees. And that's a huge range in terms of the impact on stability, all still within the spec. So what we do is we determine kind of the worst case, what would 8 degree, but we have somewhere 8 degrees is not going to be acceptable, but 5 degrees is fine if you're going to leave it there for 2 or 3 years. Okay.
Mm-hmm.
And then from cumulative excursions, you can add them, and it's all predictable. The other thing I'd add is the majority of products, the activation or the temperature sensitivity is such that it's easy to fail accelerated conditions and be fine for long term. And so part of the question is, do you do the work up front so you know this ahead of time and not, then you kind of know you're going to fail and you can justify, but still be fine for long term. So this is more the science side, then you got to deal with the regulatory side.
Mm-hmm. Well, thank you. So our audience is coming alive with some of the questions. Pearl, I think you can see these as well. The first one was if there's a loss of 4.8%, it's considered 5% by rounding according to the general chapter or not. So I guess your input on that.
All right,
I've seen the rounding question before as well.
This is when you consider, if you look at the guidance, it's not 5.0, it's 5%. So obviously that would be, so 4.8 also rounded off would give you 5%. More importantly, after rounding also, you would have to still, yes, that would be considered as a significant change only. Because here, I think the question is, it's simple, like if you have 100 and then you minus, if you would get a 4.8, then would that be considered as a significant change? But it's not 5.0 or not 5.00 in this case. So we need to be looking at decimals here. So when you round it off, it will come as 5%. So if it's anything above 4.5, that would still be considered as 5% and therefore significant change.
Okay. And there's a follow-up here. Can we confirm that the 5% change governs pH fluctuations over time, or is pH simply a significant change based on spec range acceptance criteria?
Yes, pH, as we just saw, is if it doesn't comply with the specification, then that is considered as a significant change. And more than just selecting the pathway for storage conditions or for selecting a shelf life, it's more concerning is because we need to look at what is driving this pH change and what is its impact. Because typically you have pH for, say, for a parenteral formulation, and if the pH has shifted here over stability, that is concerning, and an investigation therefore needs to be done to understand this pH change is driven by what and whether it needs to be controlled.
Okay, thank you. And then another one. This one is facing a challenge with one of our projects, which is a proton pump inhibitor that failed at accelerated 6 months conditions, passed under long-term conditions, and it also passed intermediate conditions at 6 months, waiting for 12-month data. What could you suggest? And I think that there's not a problem with that so far in my experience, but it is for China regulatory submission, which has stringent criteria. They want mostly passing data at accelerated conditions. So it doesn't sound like they're playing by the same rules we are. Have you had any experience with that? Any Pearl or the panel members?
Does China consider ICH, or if they have different regulatory requirements, then those would hold precedence over the ICH guidelines. So that we do not know exactly which guidelines govern here, because as I can see here, in China, they expect it to comply under accelerated stability conditions. So that apparently looks to be different from what has been recommended by the ICH guidelines.
Yeah, I agree. I think you have to make your case with what we have, exactly what you outlined there, what the intermediate was, and by normal standards, that's acceptable, and then see what they say in response to that. Before we get too far into our program, Pearl, could you advance one slide? Thank you. I wanted to do our little bit of a commercial announcement here. I'll take this opportunity to invite those who are new to Stability Hub to visit our website, stabilityhub.com, and check out our featured publications, regulations, events, suppliers, interactive forum, job opportunities, and more. While they are signed up for the drop-down form for our free monthly newsletter to see what's new, including announcements of programs like this. As a part of the Pharmaceutical Stability Discussion Group, PSDG for short, Stability Hub has resumed in-person PSDG meetings where attenders can spend parts of 3 days asking questions, sharing information, touring facilities, meeting vendors and learning their experiences, and networking to gain lifelong professional contacts and friends. We had a very successful meeting in May and are looking for companies in the U.S. that would like to host future meetings at their facilities. If you would like to learn more about attending or hosting such a meeting, send an email to stabilityhub.com/contact. Stability Hub is a non-for-profit information service to our industry, is operated by volunteers. If you would like to be involved more broadly in your industry and have an hour every week or two, or even a month, we could benefit from your participation. Send a note through Stability Hub contact link for more information. So we thank you for your attention to those, and we'll get back now to our questions. Thank you, Pearl. So on the screen, let's see, am I looking at the current one? Yes. When a product shows failure under accelerated conditions but complies with long-term stability requirements, what scientific factors should be evaluated before assigning or justifying this product's shelf life? Sounds very similar to the kind of things you were speaking about, Pearl. Is there anything else in there you'd like to add?
I would love to give you a one-line kind of an answer, but unfortunately, as everything related to the science we work on, it's a case by case that depends on which failure we see. Like, for example, we talked about pH here. You need to, based on the product that you're working on, you need to see what is the impact of this failure on other conditions. You would have a list of tests for that specific drug product. Does it impact the other tests? Because it's not that we only look at the results for that specific test, which is now defined by ICH as a significant change. But what is the impact on both the physical and the chemical attributes of the drug product? So you need to look at all the aspects. And again, like the example we just saw, even after extrapolation, if we get a value of say 24 months, but if another test doesn't comply at long term to that shelf life, you would have to then reduce your shelf life accordingly, such that all the tests that are relevant to that product, they comply at that. So that is how we propose the shelf life.
Okay. And is there anything that you consider significant change for viscosity? Still 5%?
Viscosity is, I do not recollect seeing the guidelines specifically give anything on viscosity. But yes, if there is a change in viscosity and that could have an impact on, say, dissolution as a test, then viscosity per se would not be deemed as a significant change. However, the influence of a change in viscosity on dissolution would then be categorized as a significant change.
Okay, thank you. I'll take another
We
one.
had, just to be careful, there's a functional side to that. Not so much dissolution, but viscosity for injectability. That you could go, we've had a couple products where the product will no longer be injectable to the same degree that it was if the viscosity goes up, and if sometimes even going down, you don't deliver the same. So depending on the type of product, most of these are liquids, for viscosity.
Mhm.
Yeah, and another point which I missed is that we talked about appearance and physical attributes. So whatever specification has been set for viscosity, if it doesn't meet that specification, then yes, that will be considered as a significant change then.
Yeah, right. That's right. And you're basically, if you're doing the thing right, you should justify the range and what is important as part of it. So, to some extent, what is significant is what is significant for that product. And it may be that it's pretty insensitive to that parameter. Congratulations.
There's another twist on the same theme, I think. What if your assay change doesn't follow linear increments or decrements? For example, at 98% initially, at 3 months, 102, 6 months, 97. So it's bouncing up and down.
Well, if you're gaining potency on a solid, that means it's your error bars in your way of measuring. That should be concerning, shouldn't it?
Yeah, I think that's why he's asking the question that's bouncing here.
If it's a solid and you're going up, you're either not sampling enough, let's say, tablets, and you're getting variability beyond what you should, so you didn't develop your method appropriately, is what it says.
So going back to the method.
Method, and it could be you need a composite that's bigger. That's statistics.
Actually, I did come across this kind of case a couple of years back, and we did receive a regulatory deficiency also on this, where the regulator said that there is a kind of variability. So
yeah, your method was no good.
yes, but in this case, it was not the method, because then similar variability was also observed because this was a generic drug product, it was also observed in the reference drug product. So the primary argument here is that this is an expected variability that is associated with the product as such, and when you compare at every interval against the initial, that value was always within 5%. It didn't exceed the percentage of a significant change. And when it was graphically plotted, that error kind of stayed within those minimal margins, as opposed to being something sporadic or random. So that was the argument that was given, and that was accepted. I think primarily because that variability was associated with the product as such, and it was not a standalone method related. But of course, that had to be justified, because as you just said, if you leave it open, that obviously that is the next thing, that is it the method that is not accurate and precise. So you need to demonstrate why you believe that that is related to the product variability, which is within that allowed range.
Yeah, I just think it's bad statistics. They should have sampled more to cut the variability such that they could tell what's a meaningful change in assay. That the fact that the reference product, somebody had the same variability doesn't change the statement that whoever did this didn't do a good job. But we wouldn't do that. I tell you, if we were doing assay, we would make sure that the content uniformity was accounted for in the size of the composite, such that assay was meaningful, or don't bother with assay.
Yeah, there's a variety of questions here. I'm looking to see which ones might be similar. Let's go back over here. Oh, why does a significant change under accelerated stability conditions not necessarily indicate that a pharmaceutical product is unstable under its proposed long-term storage? If I'm not mistaken, I think you were pointing out that sometimes 5% doesn't put it necessarily out of specifications. It's just the change itself. If you have any further comment on that, Pearl.
I'm looking at. Okay, is this just a question? Why does a significant change and is not unstable under the proposed long-term storage conditions? Am I looking at the same question?
That's the one, yes. Right
Yeah.
above.
Yes, that is what we just discussed under the scientific assessment, where we reconcile all the results. So we do not just stop with a simple statement that this significant change is only specific to accelerated stability conditions. We need to also try to understand what is the mechanism that is driving the change and how relevant that is to your real-time storage conditions. Because if you already have, say, a container that is, or you already have storage conditions where your sample is stored at a room temperature, and you have a designated excursion where it would not be possible to be exposed to higher heat conditions of 40 degrees. In that case, that can be explained that it's still stable under its long-term conditions.
Okay, and then another question, can we frame significant changes as defined in ICHQ1AR2 to biotherapeutics, i.e. the MABs? So does it change, is there application there because of the difference in the nature of the product, wider specifications, so things along those lines?
I think we just saw for biologics, it is non-compliance to the specification is considered as a significant change there.
Okay, so invoking that part of it.
Well, the other thing is, at least up to this point, they haven't accepted any kind of extrapolations or accelerated data for biologics. Hopefully, with the new guidelines, they will now start to bring in some science. But so far, that hasn't. So it's kind of irrelevant if you pass or don't pass is all you needed.
Okay, thank you. And another question from MR. Case of semi-solid preparations, the trend of stability results in case of assay doesn't follow linear. Is that a repeat of the one we just saw? Clear shelf life by extrapolation. I don't know if that's different than the one I was looking at.
Yes, I can see the same question. It's about semi-solids, right?
Yeah. I think that was similar to what you just answered on a previous one.
Yes, yes.
Okay.
Well, I'll emphasize some of this can be solved with the right statistics. If it's fluctuations because of variability, you can, populate your study suitably so that you reduce that variability by taking average.
And then there's another one from MK who's summarizing basically what we were trying to put into words before about extrapolations. In stability, we just find 5% significant change in the assay, but for other tests, what should be the criteria according to the ICH guidelines? So for the physical test, I guess that's a question I had as well. Do we have internal definitions of what constitutes a change, or we have some kind of degradation in measurement along those lines. I've not seen anybody come up with anything standard within the industry, but we have within our own operations and put in a scale of what's acceptable, what's not. So trying to match that up with something measurable, it's to be a challenge.
I mean, you can also view this in terms of a trend analysis. Are you trending to fail? In which case you can look at it as a percentage of the specification versus initial.
Mm-hmm.
And that's reasonably easy to talk about. In other words, if you've got a 50% of the specification under any condition, let's say 3 months, 40 degrees, that should be concerning.
Okay. Let's see. How long is a stability chamber excursion typically acceptable? Good question. It depends. I know everybody loves that answer, but if it's way out, it's one thing. The FDA has an official guidance that says if you're out for 24 hours, mostly for commercial products, that's reportable and needs to be investigated. But industry is usually way more stringent than that, defining what an excursion is internally. We used to have a sliding scale. If it was a high temperature, it'd only be out for 10 or 15 minutes and everybody be scrambling. If it was a room temperature one, it could be out for a couple hours and would get less attention, not quite as an emergency. So it depends. I don't think you'll find a publication that says anything other than that one guidance about 24 hours. Watching the clock here, got what time for one more? Is product assay in each interval giving different results up and down extraction process? Not sure I understand the question. It's the last one.
They're saying they have a poor analytical method. What do they do?
Go back to the drawing board, I guess, for that one. Let's see. We are getting into our last minutes here. So working with a lot of lotion and creams, I'd like to know if there is a rule of thumb for how long to wait between viscosity readings during OOS investigations for shear.
Whether you're shear thin or shear thicken is different from a question of stability.
Yeah.
To be careful.
So internally defined.
Shear is your mechanism by which it's aging, which is different from temperature and time.
We haven't got time to go too much deeper into that. Why is 2 to 8 degree temperature products not having humidity specifications? Because at that range, it's still
very important.
It's important.
That's a misunderstanding of the regulatory people.
Yeah, regulatory people has not required it. That's why.
They don't require it because they didn't realize how important it is.
Yes.
At -20, it's not important. I'll agree with that.
Okay.
But 5 degrees it is.
Very good. And Pearl, if you could go to the last slide, because it's time for that now.
Yes.
Just advance one more. There we go. Our time has flown. I'd like to take a moment to highlight our upcoming programs. Join us again on Tuesday, August 18th. That's a change date from our prior listing at 12 p.m. Eastern for a webinar on sample transport validation and control by one of our sponsors, Q1 Scientific. On Wednesday, September 9th, that same Q1 Scientific hosts a one-day in-person stability studies conference in Durham, North Carolina. Watch for announcements and sign up links in Stability Hub Newsletter and the events section of the stabilityhub.com website. As we bring our program to a conclusion today, I'd like to thank our speaker, Pearl Pereira-Nambiar, our panelists, Chris Latos, Walter Routh, and Ken Waterman, and you, our great audience, for your attendance today, and we hope to see you again soon. With that, we'll say goodbye, everyone. Thank you.